Hello and good morning, students. I am SN Ojha, and from last few days we are regular with our biology topic of class 12th. Friends, we are discussing second chapter of your 6th unit, that will be the first unit of class 12th biology, that is reproduction, sexual reproduction in flowering plants. By the time we have discussed the three important parameters, the three important topics in this chapter. First one was microsporogenesis, structure of pollen grain and other things. Second one is megasporogenesis, structure of embryo sac, what are its various components and other things. Third important topic what we have discussed is pollination, various modes of pollination which was adopted by various types of plants or especially angiospermic plants. What are the pollinating agents and how the flowers are adapted for receiving their particular male gamete and how many types of pollinations are there. In the last topic of pollination we have discussed about anemophily adaptation, hydrophily and its adaptation, entomophily and its adaptation. Some more pollinations are also there. Pollination can take place through bat, pollination can take place through birds, and all these cases we have discussed. In the previous few topics, maybe from chapter number 1 or maybe from chapter number 2, you all know that the pollination is basically of two types, that is self-pollination and cross-pollination. As far as self-pollination are there, in last topic we have discussed about autogamy, that is clistogamous flower. Some chasmogamous flower also perform autogamy. Three examples we had taken there, that is viola, that is oxalis and other plants. But now just one question arises in our brain, that the basic significance of a sexual reproduction, is the collection of variety of genes. In sexually reproducing organism, they are responsible for causing evolution. Why? Because there is a genetic vigor, genetic variability. When two parents from different genotype crosses together, they will form the progeny, and this progeny will be more adapted in the given set of conditions, and that's why sexual reproduction is evolved in all the higher plants and all the higher animals. But whenever we are talking about autogamy or self-pollination, this purpose of sexual reproduction, somewhere it is lost, because both the parent, pollen or egg, they both belongs to the same parent. The parents are genetically similar and the progeny is also genetically similar. This will cause few problems. First one is, it has some drawbacks also. So first one is, new traits are not introduced. Second one is, the progeny will lose its genetic variability, and when genetic variability is lost, definitely its adaptability, its adaptation or adaptability is also under the question mark. And the next very important is, the progeny is not true copy but is somehow similar. So what happened? The recessive genes are basically of two types, dominant and recessive. So here the recessive genes, the chances of appearance of recessive genes are more, because it always undergoes self-pollination. So, in today's topic, I would like to cover two important aspects of this chapter. The first one is outbreeding devices, and the second one is double fertilization. So, let's start today's topic. This I have discussed already, that we have discussed about the, this is what again the same flow chart what you are watching from last two videos. So the whole chapter is divided into three parts: pre-fertilization, double fertilization and post-fertilization. In pre-fertilization there are various events: microsporogenesis, megasporogenesis, and pollination and pollen pistil interaction. In double fertilization, so here for today's topic, we are going to take the outbreeding devices from pre-fertilization, outbreeding devices. Second topic we are going to cover about pollen pistil interaction, and third topic we are going to cover about the double fertilization. Okay. And after this lecture, then in further lectures we will include post-fertilization, right? So first, as I was discussing about autogamy. So in the sexually reproducing plant, when the pollen grains belongs to the same parent and when the plant undergoes continuously self-pollination, this will cause the loss of vigor, and this is called as inbreeding depression. Inbreeding depression, that is, the progenies have multiple problems. For example, if any genetic disorder which is recessive in the plants, so, and if the plant undergoes always self-pollination, so this genetic disorder, the chances of appearance of such recessive genetic disorders are more for self-pollinating plants. Moreover, genetic vigor is also lost. Genetic variability is under question mark. So these are some of the drawbacks of self-pollination, which causes inbreeding depression. For countering this inbreeding depression, few plants adopt methodologies, and that is called as outbreeding devices. That is, it is a phenomena or it is a process in which a plant rejects self-pollination or try to prevent self-pollination and enhances cross-pollination. And that's why it is called as outbreeding. Out means from outside. Outbreeding devices. Some events, some significance which are breeding outside. So there are multiple methods by which plant adopted the outbreeding methodologies, or plant promote cross-pollination and suppresses self-pollination. Let us discuss one by one about all the methods. Let me begin first, that is unisexuality, also called as declining. We all know that a bisexual plant, suppose if it is a bisexual flower, this bisexual flower has a stigma or pistil, and it has also a anther. The pollen grain from the anther transfers to the stigma. This is what is the normal process. In unisexuality or declining, what plant will do? Plant will suppress its one sexually reproducing part, either male or female. So in this process, either the anther or the male or androecium gets suppressed and could not able to produce pollen grain, or the stigma or pistil could suppress and could not able to accept the pollen grain. And that's why it is called as declining. That is, it declines one of its sex organ, either male or female. Second outbreeding device is dichogamy. Dichogamy. Again, let me make myself very clear that self-pollination can occur when pollen grain from anther transfers to the stigma of the same flower and the stigma receives this pollen grain. So here two things are very important. That the time of maturation of pollen grain and the time of maturation for or receptibility of stigma, both should be synchronous, similar, at the same time. It should occur at synchronous time. This method is suppressed here in dichogamy. So how dichogamy could be defined? Let us see the definition. Anther and stigma mature at different time of bisexual flower so as to prevent self-pollination. So here the anther and stigma, it matures at different time. The time of maturation of anther and stigma should not be synchronous. So what will happen? If the pollen grain is mature enough but the stigma is not receptible, so if the anther will fall on the stigma also, no fertilization, no pollen tube germination will take place. Same way, if the stigma is receptible and anthers are not mature enough, at that time no pollen grain of same flower will fall on the stigma. So this is called as dichogamy. Dichogamy can be of two types, what I have discussed now. One is called as protandry. One is known as protandry. Pro means previous. Andry means anther. So here what happened? The anther matures earlier. Anther mature earlier than stigma of the same flower. Their pollen grain become available to stigma of older flowers. So here the anther matures early and stigma by the time is not able to mature. So that's why it is called as protandry. Protandry, basically you can see from this diagram. Here this is the filament anther. The pollen grains are mature and pollen grains keep on shedding for the same flower or on the stigma of the same flower. But this is the stigma and this stigma is not receptible. The stigma is closed. So whatever the pollen grain will fall, it is not going to form the pollen pistil interaction, which does not lead to the formation of pollen tube, which does not lead to the double fertilization or fertilization. Second is called as protogamy. Proto again previous and gyne against gynosia. So here in the method of protogamy, the stigma matures and become receptive before anther. So anther could not able to produce or shed pollen grains, but the stigma matures before that. So what will happen? The anther is not mature, so this anther is not going to produce any pollen grain. When anther is not going to produce any pollen grain, no pollen grain will shed upon the stigma of the same flower, and this stigma can receive the pollen grain from different flowers. Okay, so that's why it is called as protogamy. Okay, so anther, as you can see here, that stigma is mature and anther is not shedding pollen grain. And here, stigma is not mature or not receptive, but anther is shedding the pollen grain. So like that, there are two important ways called as protandry and protogamy. Protandry is performed by some of the plants, for example, sunflower. And remember this flower, salvia. Salvia we have discussed in our previous topic also. Okay, there you have seen that the stigma is exposed enough in one flower, and in another flower the stigma is not so exposed enough. Okay, so that's why cross pollination. And if you miss the link, you can watch the previous videos for getting the continuity of the topics also. And protogamy, it is performed by Mirabilis jalapa. Mirabilis jalapa, very famous 4 o'clock plant. We are going to discuss this plant in our genetics topic also. Then gloriosa, then other plants are also there which performs the protogamy. Okay, so this is the second method, dichogamy. First one was declining, second one was dichogamy. Next, the third important outbreeding devices is hercogamy. In the method of hercogamy, the bisexual flower, the structure of male and female sex organs itself proves as a barrier to the self-pollination. So here there is a barrier, or they themselves prove itself as a barrier, so that because there should be a proper synchrony, right? A physical synchrony also, because the pollen grain has to shed off to the stigma. Remember in Zea mays, when we are discussing about the adaptation of anemophilous flowers, there we have discussed that the anther should present at the tip of the plant and stigma should present at the base or at the bottom, so that whenever the pollen grain will shed off, the stigma can gain the anther. But in this case what happened? A barrier is established, so that maybe in the form of a height or maybe in the form of something else, so that the pollen grain of same flower could not come in the contact with the stigma of the same flower. Now let me explain with the help of diagram. For example, this is the case of a normal flower. So what will happen in this case? Whatever the pollen grains will be formed from this anther, the pollen grain will shed off to the stigma, because the stigma and anther, they are approximately at same height. So whenever the anther will form, the chances of self-pollination is more. This is the normal flower where no hercogamy can be seen. But if you see in this or this case, and especially in this case, what is happening here? Here the stigma is too tall. And anther, the structure of anther is too short. So a pollen grain, the chances of the pollen grain to mature here and shed off and go up is very, very less. We cannot say no, there is no chances. There are chances, but the probability is very less. Okay, and that's why this is acting as a mechanical barrier. It is acting as a barrier. And due to this barrier, the stigma can receive the pollen grain from other plants, but do not able to receive the pollen grain from same plant. That's why it is called as a hercogamy, which is the third outbreeding device. Okay, next. Fourth one is, again, very interesting thing. It is called as self-incompatibility. Self-incompatibility. What do you mean by compatibility? We have discussed in chapter 1. Compatibility means every plant initiates the growth of self-pollen. The pollen tube formation is favored and of same flower, and the pollen tube from other species could not able to be favored by stigma. Here the case is little bit reverse. Okay, so self-incompatibility means pollen grain of flower do not germinate. Pollen grain of a flower do not germinate on stigma of the same flower due to presence of similar self-sterile genes. There are some genes which causes sterility also. So due to presence of same self-sterile genes, the pollen grain which belongs to same flower could not able to germinate on stigma. Okay, so for explaining that, we can understand by these three diagrams. In first case, for example, if this plant has two genes, that is called as S1, S2. As you can see here, it is the stigma of a plant having a gene S1, S2. Okay, if if the pollen grain has different genes, that is S3 and S4, a pollen tube formation takes place. The pollen tube extension takes place, and which perform the normal double fertilization. But if suppose the same plant S1, S2, these all are S1, S2 plant, S1, S2. So these all plants have a genes S1, S2, that is the stigma belongs to the plant having a gene S1, S2. And suppose if the pollen grain of, here we have discussed pollen grain S3 and S4. So S3 and S4 does not belong to the same category because it has S1, S2 and it has S3, S4. So pollen tube formation takes place. Here what happened? S1, the pollen grain of S1, and this is the pollen grain of S3. In this case what happened? Since the plant is S1, S2, the plant has genes of S1, S2, so it will promote the pollen tube extension for other genes. That is, S3 is forming a long pollen tube. S3 is forming a long pollen tube. But S1 is not forming any pollen tube. S1 is not forming any pollen tube because of the self-incompatibility, self-sterility, because the same S1 gene is present here also. Same way, if the pollen grain has the gene S1 and S2, if suppose two pollen grains, one has the gene S1, another one has the gene S2, so here also you can see that no pollen tube formation takes place because the same genes are present in the female part also. Okay, and this method is called as self-incompatibility. So let me again define it, that pollen grain of flower do not germinate on the stigma of same flower. Pollen grain of a flower do not germinate on the stigma of same flower due to presence of similar sterile genes. Sterile means non-fertile. Similar sterile genes. So wherever the sterile genes are matching, their pollen tube formation does not take place. And wherever these genes are different, then pollen tube formation must take place. This is also a very important device for promoting outbreeding devices, for promoting cross-pollination. Okay. Next, one more thing, and that is called as heterostyly. Hetero means different, and styly. Styly means style. The style, the thin long filament which connects the stigma to ovary. Okay, so that is called as the thin style. So heterostyly means there are 2 to 3 different types of flower with different height of style at stem. The height of the styles are different for the stem. So here, as you can see, in one case there is the style is too long, as you can see here. Okay, the anthers are too short and style is too long. And in other case you can see the style is too short. So anthers normally they shed off their pollen grain somewhere here and there, but the chances or probability of pollen grain to shed on this stigma is very less. Similarly, here also the anther shed off either here and there, but the chances of anther to shed off on the stigma is again very, very less. And that's why it is called as heterostyly. Heterostyly. Different styles. Right? Okay. So these all were the outbreeding devices. Let me recap it once again. The first one what we have studied is declining or unisexuality. The second one what we have discussed is dichogamy. In dichogamy we have studied about 2 components, protandry and protogyny. Third one we have discussed about Hercogamy. In hercogamy, we have seen that the height of stigma and anther both were different. The fourth one was self-incompatibility, in which self genes are not compatible and self genes are rejected and foreign genes are to be attracted for extension of pollen tube. And last one we have discussed about heterostyly, and there the style, that is the female reproductive part style, is of having the different approaches. These all methods are collectively called as outbreeding devices, and they all have a single motto, a single application, that is to reduce the chances of self pollination and to enhance the chances of cross pollination, to maintain genetic variability, genetic vigor and other things. Now, next topic we are going to study is an artificial hybridization. It's a small topic which is given in your book, but this is a very extensive and very important topic as far as Sir Gregor John Mendel is concerned. You must heard this name in your class 10th genetics. Gregor John Mendel, also known as father of genetics, and he has performed his experiment on a plant, that is garden pea plant, called as Pisum sativa. There you have studied that we in 10th class, if you remember a little bit, we have studied that a capital T crosses with a small t, or a capital R crosses with a small r, or a capital G crosses with a small g, and all these things means dominant is crossing with the recessive. And like that, hybridization, mixing technique he has given. Gregor John Mendel has used this method in a very extensive way for his experiments. That's why it is called as an artificial hybridization. Hybridization means mixing. So whenever it is natural mixing, what most of us know about, the also called as a pollination. So in the pollination there is no human interference. It is either by the nature, either by the insect or by the air, by biotic or abiotic, various agents are there, and they are performing the transfer of pollen grain. The pollen grain will extend to the pollen tube, and then the fertilization takes place. But if the same thing is done through human interference for solving a particular purpose, for lab experiments, it is termed as artificial hybridization. Artificial hybridization includes two important steps. The first one is emasculation and second one is bagging. So I have taken the same example of a same flower which was that is Pisum sativum. So if you see the flower of the Pisum sativum, it looks something like this. So what happened? What we are doing in the artificial hybridization, the first step is emasculation. Pisum sativum has a bisexual flower, we all know. It has an anther as well as it has a stigma also. So what we are doing here, we are artificially cutting and removing the anther. You can see this is the anther and this is the stigma. So in the first step, what we are doing, we are artificially cutting and removing the anther, and artificially we are converting a bisexual flower to unisexual flower. Okay, before maturation of anther or before maturation of stigma, that is important, right? So you have what you have taken. You have taken a bisexual flower. The bisexual flower has both stigma also and anther also. So anther is mechanically cut off before maturation of stigma and before maturation of anther. Now the flower has only gynoecium. Then, so this is called as emasculation. In next step, what we are doing, pollen transfer. Whatever pollen of a desired trait or whatever pollen of a desired family, we want to hybridize, we want to cross pollinate. That pollen grain is artificially collected and shaded on the stigma. Okay, when once the shedding is complete, either by the help of brush or by other equipments, in class 11 the herbarium making, you all have studied about that. So when once the artificial shedding is complete, then this flower is tied up or tagged up with a polythene bag, and that is called as bagging. So when one suppose the fertilization takes place, this is the flower, anther is removed and cut off. So when once the pollen grain of different flower of different genetic composition is shedded mechanically, after that this flower is bagged or packed in a bag, so that the further pollination could not able to take place. We are simply covering the flower by the help of polythene bag, so that the further pollination could not able to take place. And whatever desired character we want to inculcate, that character could be inculcated in the system. So that is called as artificial hybridization. Artificial hybridization. Okay. Now, next we are moving to the very important part, and just before double fertilization, it is very important to check the compatibility, as so many times we have discussed this term compatibility. Now, if you take a normal condition of pollination, now whatever outbreeding devices we have discussed, this is an extra approach to promote the things, okay, to promote the cross pollination. But normally what happens? Normally a plant having the stigma with the pollen grains, when once the pollen grain shed off to the stigma, there is a series of reaction which takes place on pollen grain and stigma. So suppose this is the pollen grain, and suppose here is the stigma. So between the pollen grain and the stigma, there are a series of reaction which takes place. These all are called as pollen pistil interaction. Here the stigma has a right to either initiate the pollen tube formation or to stop the pollen tube formation. That depends upon the species. So again, remember, in one class we have discussed, suppose there is a stigma of a hibiscus, and suppose by any chance, either by insect or by wind or by any chance, if a mango pollen grain comes and falls on the stigma of hibiscus, do you think that mango pollen grain is going to fertilize the ovule? No. Because hibiscus can take the pollen grain, can accept the pollen grain of hibiscus only. Okay, so in the same way, other species pollen tube could not able to form the pollen grain could not able to form the pollen tube, and that is called as pollen pistil interaction. When once the stigma gives the green signal that this pollen grain belongs to the same species, then only the extension of pollen tube takes place. So for example, if suppose this is the pollen grain of different species, and suppose these are the pollen grain of same species, so the pollen grain of different species can fall on the stigma because that is a physical process, but it never undergoes pollen tube formation. No biochemical process could take place because this pollen grain is rejected. But the pollen grain which belongs to the same species undergoes the extension of a pollen tube, and then it reaches to the embryo sac, which further forms fertilization, which is normally called as double fertilization. Well, once the pollen grain is accepted by the stigma, then before fertilization, As I told you that in the angiosperms, it is indirect type of pollination. Pollination, in the previous topic we have discussed two types of pollination, direct pollination and indirect pollination. Direct pollination takes place directly in the gymnosperms, where the pollen grain directly shed off to the ovule. But here it is indirect pollination. That means the pollen grain shed off at stigma and the pollen nucleus has to travel a long path because embryo sac is present below. This is termed as pollen tube extension. Just before double fertilization or just before fertilization, a pollen tube which sheds on the stigma, a pollen grain which sheds on the stigma, this pollen grain cover a long distance and it reaches to the egg which is present at the lower part by the process of formation of pollen tube. Pollen tube. Let us discuss a little bit about pollen tube and what are the important mechanisms which takes place in the pollen tube. Pollen tube is a long tubular structure which extends from the stigma by the pollen grain, and remember in pollen grain we have discussed about there are two nucleus. First one is vegetative nucleus, or first cell is called as vegetative cell, and second one is generative nucleus and the cell is called as generative cell. So here this is the pollen grain where we have discussed we have two types of cells. Okay, so there is a small generative cell and there is a large vegetative cell. So what happens? These two nucleus, the formation of a pollen tube is the function of vegetative nucleus. What will happen? The vegetative nucleus comes out through the germ pore. Exines have perforations and that perforations are called as germ pore. So exine come out through the germ, pollen tube come out through the germ pore, which is leaded by vegetative nucleus. The formation of pollen tube is the function of vegetative cell, and hence this cell is also called as tube cell, and this nucleus is also called as tube nucleus. So a pollen tube is headed by one tube nucleus, that is one vegetative nucleus, and it is followed by generative nucleus. It is followed by, this is generative nucleus and this is vegetative nucleus. Vegetative nucleus. Okay, so as in the due course of the extension of a pollen tube, this generative nucleus again split into two nuclei. Okay, so as you can see here the pollen tube is headed by, this is vegetative nucleus and it is leaded by two sperms. It is written here two sperms, that is two generative nucleus. Two generative nucleus. So in the due course of extension of pollen tube, it is headed by vegetative nucleus and the generative nucleus again undergoes division to form two vegetative nuclei, which can be seen. Right. So it has one vegetative and two generative nucleus. Moreover, the pollen tube starts secreting an enzyme which is termed as pectinase. Pectinase. Because the pollen tube has to come from the style. Style is basically composed of various types of cells, and these cells has to give the path for the pollen tube so that the pollen tube can reach from stigma to embryo sac. Okay, this pollen tube, hence the pollen tube secrete an enzyme that is termed as pectinase. The formation of pollen tube from the stigma to the ovary, this is a chemotaxic movement. That is, it follows the chemical concentration. And if you remember the structure of embryo sac, we have discussed that it is the function of synergids. Synergids have various finger-like projections, that was termed as filiform apparatus, and this filiform apparatus secretes some boron component, and these boron component makes the path for the pollen tube extension. The pollen tube extends by following the concentration of this chemical component. Moreover, this path is also guided by, the polar tube path is also guided by arbitrators. When the arbitrators guide the path, it comes to the polar grain extension, polar tube extension. Slowly, the polar tube reaches from stigma, and now it comes to the embryo sac. It comes to the ovary, right? Well, in the ovary, a polar tube has to penetrate the wall of ovary, or has to pass the ovary. Ovary structure we have already, megasporangium, we have already discussed about various components, nucellus, integument, micropyle, chalaza, all these things. So it has to cross the structure and reaches to the central part where the egg is present. Okay, this process takes place by three processes. Either the pollen tube can enter through micropyle, or the pollen tube can enter through chalaza, or the pollen tube can enter through integument. So what is the path of entrance of pollen tube? On the basis of that, the fusion is of three types. First one is termed as progamy. If you remember, this is the embryo sac, it is the chalazal pole, this is the micropylar end. So progamy is a normal method through which most of the fertilization takes place. Okay, so it is the process in which pollen tube enters into the embryo sac through micropyle. As you can see, this is the pollen tube. Here, this is the pollen tube, and this pollen tube is entering into the embryo sac through micropyle. Hence, it is called as progamy. Another one is called as mesogamy. Sorry, here a little bit across is there. So mesogamy. Sorry, this one. Mesogamy. So this is mesogamy, and this is chalazogamy. So in mesogamy, what happened? The pollen tube enters through integuments. This is again the pollen tube. This is the pollen tube, and it penetrates the integument and reaches to the embryo sac. The third one is chalazogamy. In chalazogamy, as you can see, this is the pollen tube, and the pollen tube enters through the chalaza, because this end is called as the chalaza. So on the basis of from where the pollen tube enters into the embryo sac, again the fusion is of three types, that is progamy through micropyle, that is mesogamy through integument, and that is chalazogamy through chalaza end. And you know that micropyle and chalaza both ends are situated totally opposite to each other. Okay. Now, when once the pollen tube formation takes place. Okay, let me come from first. Now pollen pistil interaction is done. Pollen tube extension took place. Pollen tube now enters into the embryo sac. Second, and now the event begins, that is called as double fertilization. Double fertilization. A characteristic feature of all the angiosperms are called as double fertilization. Why it is called as double fertilization? A little bit idea you have, because in class 10th also you have studied. Here the two male gametes, remember, one vegetative nucleus which is followed by two male nucleus, two generative nucleus. These two generative nucleus is going to fuse two different cells at the same time, and hence it is called as double fertilization. So one fusion going to take place at egg. The fusion of one male nuclei to egg is called as syngamy, and the second fusion going to take place at polar cell, and in the polar cell, central cell, and in the central cell or polar cell, one male nuclei fuse, and this fusion is called as triple fusion. So in this way, there are two important events which takes place in double fertilization, which is a characteristic feature of angiospermic plants. Let us see. In syngamy, it is the process of fusion. Now, it is the process of fusion of one male nucleus to the egg. So, one male nucleus having genetic constant haploid, when it fuses with egg, again that is haploid. So, haploid plus haploid, this will form a diploid, and diploid cell, and this diploid cell is called as zygote. Zygote. This zygote is diploid, which undergoes mitotic division to form embryo. So, embryo formation is the characteristic, is the result of syngamy. On other hand, when we discuss about triple fusion, why it is called as triple fusion? Remember, in the central cell, there were two nucleus called as N plus N. N plus N. When these two N plus N fuses with a male nucleus, second male nucleus. So what will happen? These three unite to form, that is called as 3N cell. N plus N plus N. 3N. And this 3N will form primary endosperm nucleus. Primary endosperm nucleus. This primary endosperm nucleus forms primary endosperm cell. And this primary endosperm cell later on divide to form endosperm. Okay, so that's why there are two methods, syngamy and triple fusion. So let me recap here. In syngamy, one male nucleus fuses with egg to form a diploid zygote, which later on modified to form embryo. In triple fusion, two male nuclei fuse with another male cell, another male nuclei to form a triploid nucleus. Triploid nucleus. And this triploid nucleus is termed as primary endosperm nucleus, which later on form primary endosperm cell. So, as you can see here, when both the nuclei are releases, so one nuclei is fusing with the egg, and another nuclei is fusing with the polar cell or central cell. As a result, this will form an endosperm. This endosperm is of 3N, and this will form a zygote. This zygote is of 2N, and hence it is termed as double fertilization. Okay. Now, little bit more if we highlight on the double fertilization. Here, what happened when the male nuclei release? So, male nuclei releases from which part? So, definitely it will releases from through the synergid, because synergid is only secreting the boron related compound, and the polar tube will follow the concentration of a boron. Okay, so when the polar tube reaches to the embryo sac, it enters through the micropyle or chalaza or integument, whatever it is. So it reaches to the synergid, and as while passing, this is the synergid, and this is the filiform apparatus, and this is the polar tube. So as the polar tube enters into the synergid, this releases its two cells, two nuclei. As it passes through the synergid undergoes degeneration. This you can see here, it is degenerated synergid. Okay, and as a result, one male nuclei fuses with fuses with egg. This will form, after fusion, this will form zygote, and this process is called as syngamy. And other male nucleus reaches to polar nuclei, and this is called as triple fusion, and which results in primary endosperm nucleus, which is diploid. Here zygote is diploid. Okay, so this is what is the exact mechanism how the double fertilization takes place and what happened to the synergy. Okay. Well, so this is all about the outbreeding devices and double fertilization in the angiospermic plant. In next topic, we are going to discuss what is the scenario of an embryo sac after fertilization and what are the important events of post fertilization when once the fertilization is over. Till that time, take care of yourself and keep revising. In case of any doubt must be there. In case of any doubt, please consult to your respective teachers. Thank you very much.